Right, so we are going to talk about the cell membrane today. Last time, if I'm not wrong, we started on cytology. We talked about the nucleus and also the cytoplasm. So today we are going to talk about the cell membrane. We are going to focus on the structure and functions of the cell membrane. To be more specific, these are the things we're going to learn. We are going to look at the structure of the cell membrane. We are going to state the major functions of the cell membrane. We are going to look at the biomolecules which are on the cell membrane and perhaps try to see the proportions in terms of percentages they contribute in the cell membrane. We are going to narrow down to the proteins on the cell membrane. We are going to see how we classify them and their major roles. And lastly, we are going to describe how molecules are transported across the cell membrane. This is going to apply for both small molecules as well as large particles. So let's begin by agreeing on what we had agreed before for those who weren't in the lecture last week when we started on cytology. We said that the cell consists of three components: the cell membrane, the cytoplasm, and the nucleus. In our previous lecture, we talked about the cytoplasm as having cytosol, cytoskeleton, and cellular organelles. And we stated the role of those three components of the cytoplasm. For the nucleus, we said it has three key functions: controlling activities of the cell, housing the nucleolus, which produces ribosomal RNAs, and that the nucleus within the nucleoplasm contain the genetic material, which we went ahead and also talked about the organization of the nuclear genetic material. So we completed our story concerning the nucleus and the cytoplasm, but now we have not talked about the cell membrane. So today we are focusing on the cell membrane. This lecture will be spread over 2 or 3 series. So we're just starting it today, cell membrane, and we'll still continue with the story of the cell membrane tomorrow during physiology time. What are the functions of the cell membrane? The cell membrane compartmentalizes and protects the cellular contents. This is to mean that it puts the contents of the cell in one compartment. Apart from putting them in one compartment, it also protects the components of the cell. The cell membrane is also important for intercellular communications. This is to mean two cells were to communicate within the body. That communication can be affected by the cell membrane, and especially the cell membrane of the recipient cell, the cell membrane of the target cell. That is usually through the fact that the cell membrane contains receptors for particular ligands. Let's call them hormones, for example. So those chemical messages bind on receptors on the cell membrane of the target cell. Through that, communication is achieved. But another better example is with regard to how signals are transmitted along the cell membrane of neurons. That is still a form of communication. Cell membrane is also responsible for transport of substances into and out of the cell. We are going to see how substances are transported into and out of the cell. Lastly, cell membrane contains some chemicals that we can call antigens. These antigens are responsible for identity of the cell. And this is more of immunological identity or immunological recognition. I am glad that you people are doing immunology and so you understand the term antigen. For example, when you say you are blood group A, it means that the cell membrane of your red blood cells have an antigen called antigen A. If your blood group B, the same. The cell membrane of your red blood cells have an antigen called antigen B. If you're AB, it means you have both antigen A and antigen B. Now, don't make assumption that now if you're blood group O, you have antigen O. That O actually means 0. To mean you don't have those ABO antigens, you don't have them. Right. These are the functions of the cell membrane. What is the basic structure of the cell membrane? Let's go slowly here. The cell membrane is described as being a phospholipid bilayer structure. What does that mean? It means that you have 2 layers of phospholipid. A phospholipid is something with a phosphate group, those rounded knobs, and a fatty acid chain, those 2 strings. So a molecule that contains fatty acid chains and a phosphate group is termed a phospholipid. In the cell membrane, we have 2 layers of phospholipids. This is one layer of that, and this is the other layer of that. And so they exist in form of 2 layers of phospholipids. That's the basic structure. Now, how are these phospholipids organized? Look at this. They are organized in such a way that the fatty acid chains are sandwiched between the phosphate groups. Having said so, the fatty acid chains which are sandwiched are actually hidden away from watery environment. This is a watery environment out of the cell, and this is watery environment within the cell. The fatty acid chains are hidden away from the watery environment. Because the fatty acid chains are hidden out of the watery environment, this region of the cell membrane is therefore described as being hydrophobic. However, the phosphate groups are exposed to the watery environments. This is phosphate group, this is watery environment. So the phosphate groups are exposed to the watery environment. So these regions of the cell membrane are described as being hydrophilic. They like water. Apart from phospholipids, the cell membrane also contains other biochemical molecules. The biochemical molecules which are located in the cell membrane include proteins. As a matter of fact, even though we have said that the cell membrane is a phospholipid bilayer structure, proteins actually constitute the largest amount of biomolecules on the cell membrane. About 55% of biomolecules on the cell membrane are proteins in nature. Then other lipids, I'm using the term other lipids because phospholipids are also lipids. Or let me say this, apart from proteins, we also have lipids in the cell membrane. Lipids constitute about 42% of biomolecules in the cell membrane. There are 3 major categories of lipids in the cell membrane. One of them is the phospholipids that we've already talked about. The second category of lipids on the cell membrane are cholesterol. And the third category are glycolipids. The term glycolipid means that it is a lipid combined with carbohydrate, glyco, for the carbohydrate. So glycolipids, phospholipids, and cholesterol. Those are lipids within the cell membrane. In total, they constitute about 42%. Lastly, we have carbohydrates. Carbohydrates constitute about 3% of membrane biomolecules. Most of these carbohydrates do not really stand alone. They are combined with proteins, like this manner, to form what we call glycoproteins. They tend to be the ones which contribute to the antigenic functions of the cell membrane. Right, we have mentioned that proteins contribute the largest amount of cell membrane biomolecules, 55%. I want us to narrow down onto this. We can classify cell membrane proteins in two ways. There's anatomical classification and there is a functional classification. The anatomical classification of the cell membrane proteins recognizes the location of these proteins. There are some proteins which are within the membrane, like this one, and this one. At least part of them is within the membrane. Such proteins are termed integral proteins. Among the integral proteins, there are those proteins which actually go through the whole membrane, like that and that. You compare that with this one. So among the integral proteins, the ones which actually go through the whole membrane are termed transmembrane proteins. Transmembrane proteins are therefore examples of integral proteins. On the other hand, there are some proteins which are not in the membrane, like this one or that one. They are not within the cell membrane. These ones are termed as peripheral proteins. Right. I've seen that my network has tipped a bit, so let me just repeat what I've just said. I've said that there are some proteins which are not within the membrane. Those proteins are like this one and that one. Such proteins are termed as peripheral proteins. They are not within the membrane. So those are the two categories of proteins when we classify them anatomically. This is based on where they are found within the membrane. But there's also another way in which we can classify these proteins. We can classify them based on their functions. And so we call that functional classification of cell membrane proteins. This type of classification would therefore recognize about 5 or 6 varieties of cell membrane proteins. This is purely based on function. Some of these proteins function as receptors. Now, what's a receptor? A receptor is a molecule where a chemical messenger binds. So it contains the binding site of a chemical messenger. Let's assume that chemical messenger is a hormone. For example, maybe this one. So this is a hormone. It comes and binds there. If this protein contains a binding site for a hormone, then this chemical is a receptor. Some of the proteins in the cell membrane are receptors. Some of the proteins in the cell membrane are enzymes. What do enzymes do? Enzymes catalyze biochemical reactions. So it could be a protein, yes, but it catalyzes biochemical reactions. It is an enzyme. Some of these proteins are transport proteins. And there are two categories of transport protein. When we say transport protein, it means that they facilitate movement of substances across the cell membrane. There are two categories of transport proteins. There are those transport proteins which we will classify as being ion channels. It means that they have a lumen that allow passage of small molecules. Those small molecules are usually very selective. So ion channels for sodium would be different from channels for potassium. But either way, they allow movement of small molecules through a lumen that is selective. Those are ion channels. Ion channels are different from what we call carrier proteins. Carrier proteins also transport molecules. They may not necessarily be ion channels, but they transport molecules as well. So there are two categories of transport proteins. There are those that we call ion channels, and then those that we call carrier proteins. I'll be showing you examples of carrier proteins shortly. The fourth category or fifth category, if we consider that the transport proteins are of two types, then the next category of functions of cell membrane proteins are those ones we consider as being structural proteins. Structural proteins contribute to the structural integrity of the cell membrane. They just contribute to the structural integrity of the cell membrane. So we call them structural proteins, like these ones. They are contributing the structural integrity of the cell membrane. The last class, depending on how you're counting, could be 5 or 6. But the last category are the antigenic proteins. Proteins which are antigenic in nature. In immunology, you must have defined antigen already. If not yet, I can define for you. Antigen is something that induces immunological response. So some of these proteins are antigenic, which means they can induce an immunologic response. Great. Perhaps what is important also to note is that most of the cell membrane proteins will have multiple functions. A protein can be a receptor, but it is still a transport protein at the same time. It is still an ion channel. Or it could be an enzyme, but also a structural protein at the same time. Most proteins in the cell membrane will have multiple functions. You can have a protein that has 2 or even 3 functions. So this classification is just based on the role these proteins play in the cell membrane. But a single protein can have multiple functions. Actually, that's a common pattern. So those are cell membrane proteins in terms of how we classify them anatomically as well as functionally. We agreed that one of the key functions of the cell membrane is to transport substances across it. I therefore want to take this chance to teach us the mechanisms through which molecules are transported across the cell membrane. This is easier done when we look at transport of small molecules separate from transport of large molecules, because the mechanisms of transporting them are different. So let's begin by looking at transport of small molecules. When you talk of transport of small molecules, we are referring to things like sodium ions, potassium ions, calcium ions, chloride ions, and the like, the small molecules we are talking about. So there are two major ways in which small molecules are transported across the cell membrane. One of those major ways is what we call passive transport. What do we call passive transport? Passive transport is transport of small molecules along their concentration gradients. This is to mean that the molecule is moving from a region that it is highly concentrated, that particular molecule has a higher concentration, to a region where that particular molecule has a lower concentration. So it will be moving along its concentration gradient. For example, here, this particular molecule in question here is more on this side compared to this side. So what happens with the time? With the time, we see this molecule is moving to this region until an equilibrium is achieved. When molecules move along their concentration gradient, you call that passive transport. So it means that in passive transport, metabolic energy is not really required because the movement is along the gradient. So there is no need for metabolic energy. This is also called diffusion. This is what we call diffusion in high school. But today, I want us to elaborate on this passive transport further and have some better understanding of the mechanisms that are present in passive transport. There are three forms of passive transport. This is what we call simple diffusion. This is what you learned in high school. That, if you remember that definition, just put it here. That is simple diffusion. But simple diffusion can occur direct through the membrane or can occur through an ion channel like this one. As long as the molecule in question is concentrated more on one side than another, there'll be net movement. It can move through the membrane like this if it's able to move through this fatty layer, or it can move through ion channel. That is simple diffusion. There is something called facilitated diffusion. So in facilitated diffusion, yes, the molecule in question is moving along its concentration gradient. So let's say it is more here than here. So the molecule in question is moving along its concentration gradient. But for some reason, that particular molecule may not easily pass through the cell membrane, maybe because of the chemical nature of that molecule. It might need some molecule to carry it, to push it through along its concentration gradient. When molecules are pushed by something else, but along their concentration gradient, then you call that facilitated diffusion. It is diffusing, but it's being helped to diffuse through. The third type of passive transport is what we called osmosis. I believe all of us still remember the definition of osmosis. The only thing I want to adjust in that definition is that let's look at it in terms of movement of water molecules. And let's look at it in terms of this, whatever you call hypertonic or hypotonic, let's look at it with focus on the concentration of water molecules. You therefore realize that if a solution is hypertonic, it means that it contains less of water molecules and more of solids. That's why it's hypertonic. If a solution is hypotonic, then it contains more water molecules and less solutes. So it's hypotonic. Therefore, water molecules will then move from a solution that's hypotonic, which means there are more water molecules there, to a solution that's hypertonic, which means there are less water molecules there. In so speaking, we are still having diffusion of water molecules from where they are more hypotonic to where they are less hypertonic. Osmosis is still a type of passive transport. So these three do not really require metabolic energy because the movement is along the concentration gradient. So they are forms of passive transport. Let's compare that with active transport. So in active transport, the molecule in question is being transported against its concentration gradient. If you have to achieve that, then you must use some force. It's like pushing something heavy up the hill. You must use some force. Metabolic energy is definitely required. If you have to move a molecule from where it is more to where it is less, you don't need energy. That would be diffusion. But if you have to move a molecule from where it is less to where it is more, you need some energy. That would be active transport. When we talk about active transport, we want to know the number of molecules which are being transported. That makes us then classify active transport into primary and secondary active transport. When we talk primary active transport, means that we are moving only one molecule. Transport of one molecule against its concentration gradient will constitute what we call primary active transport. However, if we are moving more than one molecule, then we'll call it secondary active transport. If you're moving more than one molecule against their concentration gradients, we want to know whether these two molecules or these multiple molecules are moving in the same direction, like this one, or they're moving in opposite directions, like these two. If they're moving in the same direction, then we use the term symptots. If they're moving in different directions, then you term that as antipodes. Secondary active transport can either be a symptot, both molecules are moving in the same direction. Either they're both moving into the cell or they're both moving out of the cell. So we call that symptot for same direction and antipode if they're moving opposite direction. One is moving in, the other is moving out. That is active transport. Now, we've been just talking about small molecules being transported, but we haven't said much about the channels that transport them. Channels which transport small molecules across the cell membrane are known as ion channels. There are three categories of ion channels on the cell. There are those ion channels which we call leaky channels. What is a leaky channel? A leaky channel is an ion channel that is always open. the channel is always open. So understand that if the channel is always open, then it means that it will always be allowing a molecule to pass through it in the direction along the concentration gradient. For example, if it's a leaky channel for potassium, it means that it will be constantly allowing potassium to move from a region where potassium is highly concentrated to a region where potassium is less concentrated. If it's a sodium leaky channel, it means that it will be allowing sodium molecules to move from a region where sodium is highly concentrated to a region where sodium is less concentrated. And the same applies to calcium leaky channels. From this lecture, I want you to understand that we have leaky channels which are always open, and that means that they always allow molecules to move through them along their concentration. What is important to note is that these ion channels are very specific. So potassium leaky channels will just be for potassium. Sodium leaky channels will just be for sodium, and the like. Now, leaky channels are a bit different from what we call gated channels. From the word gate. Remember, gates are meant to be closed, but they're not permanently closed. So, gated channels are usually closed, but they can be opened through a trigger. We then classify gated channels based on what triggers them to open them. There are some gated channels which are usually closed, yes, but they open up if there is electrical disturbance on the cell membrane. So like this green one, I hope it's green. We can see it's usually closed, but if there's some electrical change, compare this charge with this one, it means that there's some electrical change between this membrane and this membrane. If that electrical disturbance reaches a particular threshold, and the threshold will be different depending on which kind of channel. So if there is electrical disturbance that reaches threshold, those channels open. Such channels will then be called voltage gated channel. It means that they open after the voltage of the cell membrane reach a particular threshold. Tomorrow we'll be learning that there are some voltages which are on the cell membrane. Right, come to this one. There are some channels which are always closed, but they open if a ligand binds on them. A ligand is a chemical messenger. That chemical messenger can be from outside the cell or can be from within the cell. Any chemical, okay, some chemical messengers, when they bind to their receptors, so like that means that this is a channel, but it's also a receptor protein. So when that chemical messenger binds to the receptor, it will make that receptor to open its channel, like this one. This one binds there, the channel opens. It was originally closed. This one binds there, when it binds, the channel opens. So channels which are opened by a chemical messenger are known as ligand-gated channels. That is the second type of gated channels we're talking about. The third type of gated channels are channels, not in these images, but they are channels which are opened by mechanical disturbance. If there is some mechanical disturbance on the cell membrane that reaches a particular threshold, it may open some channels. So we call those channels mechanically gated channels. It explains why if someone slaps you, you can feel pain. There is a mechanical disturbance on the cell membrane of those things that are detecting that particular sensation of pain. So, 3 types of gated channels: voltage gated channels, ligand gated channels, and mechanically gated channels. The third type of ion channels on the cell membrane are what we call ionic pumps. Ionic pumps are actually mechanisms of active transport. They are molecules that pump molecules, or rather they are proteins that pump molecules against their concentration gradients. A good example, and we'll be talking about this more tomorrow, is what we call the sodium-potassium pump. That particular pump is usually an enzyme at the same time. So some books will call it sodium-potassium ATPase. It's an enzyme that breaks down ATP. So sodium-potassium ATPase. You can just call it sodium-potassium pump. A common molecule, we'll be talking about it more tomorrow. 3 categories of ion channels. Leaky channels are always open. So they'll allow molecules to just diffuse along their concentration gradient. Gated channels are usually closed, but they can be opened through a trigger. Can be voltage, can be chemical messenger, can be mechanical disturbance. Ionic pumps transport proteins against their concentration gradient using energy. There are types of active transport mechanisms. Right, we can now talk about transport of large molecules. How are large molecules transported across the cell membrane? For you to transport a large molecule, and when you talk of large molecules, we have in mind things like microorganisms. Maybe there's a bacteria that has entered your circulation. Then it needs to enter a particular cell. Maybe it's the plasmodium thing. It needs to enter the cell. How will these large molecules enter the cell? It will not be through diffusion. It will not be through active transport. How will it happen? For a large molecule to be transported across the cell membrane, it requires that a vesicle will form around that particular particle. For example, here, if this large molecule comes here, a vesicle must be formed around that particular particle, like in this manner. So the structure of this vesicle is actually the same as the structure of the cell membrane. That means some part of the cell membrane actually bud off to enclose that structure, to form a vesicle around it like this. There are 2 types of transport of large molecules based on the direction of movement of that molecule. If the large particle is moving from inside the cell to out of the cell, then we call that exocytosis. In exocytosis, the cell will be expelling the contents of the vesicle into the extracellular space. That is called exocytosis. The cell expels the contents of the vesicle into the extracellular space. If the molecule is entering the cell, then we call it endocytosis. So in endocytosis, the large molecule is moving into the cell. We classify endocytosis based on the consistency of what is moving into the cell. If that large particle moving into the cell is fluidy in nature, then we call that pinocytosis. Pinocytosis means that the cell is drinking, in quotes, because what is going in is actually fluid. The way you drink porridge, you take in fluid. Pinocytosis. That is different from when you eat ugali. If what is going in is a solid particle, then you call that phagocytosis. The cell is eating. Phagocytosis. Phagocytosis and pinocytosis are examples of endocytosis. Right. I think I should be done now. You should be able to answer these questions that are projected right now. You will not answer them now. I believe you can say what they are. All right. So we'll stop there. Thank you very much. Tomorrow, we will pick from there. We'll be talking about how cells communicate with each other. We'll also talk about body fluid compartments. And we will also begin talking about cell membrane potentials, electrical activities on the cell membrane. Right, so we can stop there. Have a nice evening. If there's no question, or rather, if there's a question, you can ask. If there's a question,